Welcome to this lecture on animal biotechnology. Today, we are going to explore one of the most fascinating and powerful tools in genetic engineering, known as zinc finger nucleases. If you have ever wondered how scientists can go into a living cell and fix a single genetic mutation without damaging the rest of the DNA, you are in the right place. Let us dive in and understand how these molecular scissors work. Before we look at the structure of these molecules, let us understand the problem they solve. Imagine you have a massive encyclopedia with billions of letters, and you need to find one specific typo to fix it. If you just use a random pair of scissors, you will cut the book to shreds and destroy important information. Instead, you need a highly trained sniffer dog that can find the exact misspelled word, carrying a tiny pair of scissors to make one precise cut. In the world of genetics, zinc finger nucleases act exactly like this sniffer dog and scissors combination. Now that we understand what they do, let us look at how they are built. Formally, a zinc finger nuclease is an engineered artificial restriction enzyme designed to target and cleave specific DNA sequences. It is built by fusing two distinct protein domains together into a single molecular machine. The first part is the zinc finger DNA binding domain, which acts as the guidance system that recognizes and binds to a specific sequence of DNA nucleotides. The second part is the DNA cleavage domain, typically derived from a bacterial enzyme called FOC1, which acts as the molecular scissors to cut the DNA strand. Understanding the structure leads us to a very important functional requirement. Because the FOC1 enzyme only works when two of them come together, zinc finger nucleases cannot work alone. They must be designed in pairs that bind to opposite strands of the DNA. When both the left and right nucleases bind correctly to their target sequences, the FOC1 domains dimerize, or join together. This dimerization is what activates the scissors, creating a double-strand break at the exact desired location. This two-part verification ensures that the DNA is only cut when both guides find their exact matching sequences. So, why does this precise cutting mechanism matter so much in biotechnology and medicine? Before these nucleases were invented, scientists had very little control over exactly where a new piece of DNA would insert itself into a genome. Random insertion could accidentally disrupt healthy genes and even cause cancer. These tools revolutionize genetic engineering by allowing researchers to perform targeted gene editing. This precision is absolutely critical for gene therapy, where we want to fix a broken gene causing a disease like sickle cell anemia without harming the rest of the patient's DNA. They are also vital for agricultural improvements. To truly appreciate how these tools achieve such incredible precision, we have to look at the mathematics of DNA sequence probability. Since DNA is made of 4 bases, we must ensure our target sequence is long enough that it does not appear anywhere else by accident. This brings us to our probability formula. P equals n times 1/4 raised to the power of L. Here, P represents the expected number of times a specific sequence will appear by random chance. N is the total size of the genome, which is about 3 billion for humans. L is the length of the target sequence. As L increases, the fraction becomes incredibly small, meaning the expected number of random matches drops dramatically. Now that we have our formula, let us see this with a concrete example. Suppose you are designing a nuclease pair to knock out a faulty gene in a mouse model. The mouse genome has about 3 billion base pairs. You use a left and right nuclease with 4 zinc fingers each, making 8 fingers total. Since each finger recognizes 3 base pairs, the total length L is 24. We plug N equals 3 billion and L equals 24 into our equation. Calculating this out, we find P is approximately 0.00001. This incredibly small number means there is virtually zero chance this sequence exists anywhere else by random chance, making our design highly safe. Having seen how to calculate the probability for a known length, we can now reverse the process to prove what the minimum safe sequence length must be. This is a crucial derivation for any genetic engineer. We want our molecular scissors to cut at exactly one unique location in the entire human genome. Therefore, we set the expected number of random matches, p, to exactly 1. We know the human genome size, n, is 3 billion base pairs. By substituting these values into our probability equation, we set up the foundation to solve for the unknown exponent L. This will give us the exact threshold where a sequence appears only once. Continuing with our derivation, our primary goal now is to isolate the term containing the variable L. By rearranging the equation, we move the fraction to the other side, showing that 4 raised to the power of L must equal 3 billion. This makes intuitive sense because each position has 4 possible bases, and we need enough combinations to exceed the total genome size. To solve for an unknown exponent, we must use mathematical logarithms. We take the base 10 logarithm of both sides of the equation. Using the properties of logarithms, we can bring the exponent L down to a solvable level, multiplying it by the log of 4. Now we arrive at the final steps of our derivation. We divide the log of 3 billion by the log of 4. Calculating this gives us a value for L of approximately 15.8. However, we cannot physically have a fraction of a DNA base pair in reality. Therefore, we must round this up to the next whole number, which is 16. We have just mathematically proven that a DNA sequence must be at least 16 base pairs long to be statistically unique in the human genome. This rigorous mathematical approach is what prevents dangerous off-target cuts and keeps modified cells healthy. So we have proven that a length of 16 base pairs is required. This brings us to how we actually design the nuclease pair in the laboratory. Because of their biological structure, each individual zinc finger domain recognizes exactly 3 base pairs of DNA. If we design a pair using 3 fingers on the left and 3 fingers on the right, they will recognize a total of 18 base pairs. Since 18 is greater than our mathematically proven minimum of 16, this design is more than enough to guarantee a unique cut. This ensures the patient remains completely safe from off-target mutations. Let us bring all these concepts together and look at this process visually. In this diagram, you can see the double-stranded DNA molecule running horizontally. Notice the left and right zinc finger nucleases binding to opposite strands. The blue and green ovals represent the zinc finger domains, with each oval binding exactly 3 base pairs. In the center, the pink and orange circles represent the FOC1 cleavage domains. Because the left and right guides have found their specific targets, the 2 FOC1 domains have come together to form a dimer. The yellow lightning bolt shows the exact moment of cleavage, creating a precise double-strand break right where we want it. We have covered a lot of ground today, so let us summarize the most important ideas. First, zinc finger nucleases are powerful molecular scissors that combine a programmable DNA binding domain with a DNA cutting enzyme. Second, they must function in pairs, which acts as a safety mechanism to ensure precise double-strand breaks. Finally, by understanding the mathematical probability of DNA sequences, we can design these tools to target sequences long enough to be absolutely unique in the genome. This rigorous precision is what makes modern genetic engineering and safe gene therapy possible. You now have a solid understanding of how zinc finger nucleases work.